At Kapelica Gallery we present cellF, the world's first neural synthesizer. It's a collaborative work from 7 people. Basically what we have made is an interface that has a human brain network growing in a temperature controlled environment. At the top, you see at the top in the tower, this human network, brain network, is constantly pushing out neural activity as synapses or action potentials. And we're taking this data as electronic signals and putting it through a modular synthesizer. And the modular synthesizer shapes and adds color to these electrical signals, and then these shaped and colored signals are placed out into the space as more of a musical kind of sound. The neural synthesizer, we call cellF, is also able to respond to human musicians. So in a performative setting, not only in a gallery setting, we're able to produce a live interaction between humans and the instrument with the brain growing inside it. So how we do this is we have human musicians producing sound through their instruments, be it voice or guitar or anything that produces a sound, and we have that streaming into the neural network via frequency switches. So the sensors are able to be tuned, so when a particular artist produces a particular note, we can send that to any part of the neural network as a stimulation. So this really talks to and actually comments about how neural plasticity can be influenced and changed over time due to the stimulations that it gets. The neurons are actually iPS-derived neurons, and what that means is the artist, Guy Ben-Ary, took a skin graft from his wrist and reverse engineered those skin cells to become stem cells. And this is a method that's still quite cutting edge, but is now about 7 or 8 years old. And when these stem cells are produced, of course, we're able to move stem cells along the lineage of many other kinds of human cell types. So for this artwork, Guy Ben-Ary has taken the stem cells from his skin graft and differentiated them to human neurons. And those neurons are then grown on an electrode-based petri dish that has 64 electrodes. And basically what those 64 electrodes are kind of like open wires that actually we use as an input-output or a read-write device. So the neurons are grown on the electrodes. The electrodes are broken out to the front panel where you see the black front panel. And the lights that are flashing there show particular activity in certain parts of the network. The neurons are able to survive almost indefinitely, but they actually need to be fed every 2 days. Yes, so we've turned skin into brain. So the actual physical form of the device is actually after lots of consideration between Guy and I. We talked about many things that influenced us, and also about how to really talk about what is happening at a microscopic level. If you can see the front of it, it's actually a smooth silver-based cone that kind of looks like a loudspeaker. And I definitely have influence from the early loudspeaker technology, and also a lot of really a lot of lab-based machines that were used in early music recording. So the color palette was particularly chosen for this reason. The form itself, we have a central column of black in the middle that you see, and that initiates and really describes what is happening. The source of the electronic activity is from the brain itself at the top, and it's brought down and amplified and taken through to the modular synthesizers. But the actual physical form is that the black tower pierces through the silver smooth lining and runs around the back, kind of like an amplified horn. But when it moves around the back, it comes out to the front, and we find that the genesis of the sound is actually front and center of the speaker or the output. And this is quite important because basically what we're building here is a feedback device. We're building a machine or semi-living machine that is able to listen to human musicians and respond. So it's kind of a feedback that would happen in a live jam session in a way. And this is indicative in the design and physical design of the device. We have the loudspeaker in front of the genesis. It's like holding a microphone in front of a loudspeaker. You have a feedback kind of form. Also, on a microscopic level, this is exactly what is happening. So we're taking the millivolts from the neurons, and we're amplifying them in an analog realm. There is no computers, there is no microprocessors, there's no coding in this device. It's all purely analog voltages, amplified and attenuated. So we take the millivolts from the neurons, amplify it out through the modular synthesizer, pass it out to the speakers produce a sound, the human musician hears the sound, responds with their instruments. That is taken as analog voltage, placed back into the network as stimulations, alters the activity of the neural network, and thus a feedback or reaction sound is played out in the speakers in the space. So it's a purely wet-alogue instrument that is very unique and always changing. Although we do have a completely working lab in this artwork, it is quite difficult to use because it's a little bit crowded. One of the really great things about working here at Kapelica is that there is a functioning biolab in the gallery itself. And I think that's very unique. I think this is the only place in the world, as far as I know, that has an art gallery and a biological wet lab in it. So, like I said, although we can produce most of our work in the artwork itself, it's really handy for us to have all of the machines and facilities here at the gallery to produce the neurons and also to sustain them and keep them alive over the course of the installation. The incubator at the top is a temperature-controlled environment. It's 37.5 degrees Celsius with 5% CO2, and this environment keeps the brain neurons happy and able to take stimulations and produce these action potentials. Around the back, if you can see, there is a sterile hood where we are able to feed the neurons. Every 2 days they need to be refed with proteins and amino acids and a special recipe of brain food. And we're able to situate this artwork inside a gallery and actually produce live compositions of sound that are directly related to the neural activity at the dish. And we're also able to culture and produce multiple brains over the course of an installation in this very device. This is an artwork that has been produced as a collaborative work, instigated and initiated by Guy Ben-Ary and Darren. I'm involved with the design and fabrication of the device. Andrew Fitch is responsible for all of the electronics, and we have 3 scientists, neural engineers that have helped us, Stuart, Hodgetts, Bakkum and Edel. We've worked intently on this work for almost 6 years now, I think. And now it's traveling through Europe and also hopefully to Northern Hemisphere countries to perform with local musicians and produce a live jam session that is unique for every space. So it's a unique piece that is always changing. And the device itself is actually a working lab.